DNA Depot presents Foods as Science Tools, Kit DD205. The objective of this experiment is to determine that certain food components can act as biological indicators and be useful biological tools as components. Your kit should include dry yeast, starch, sugar, iodine solution, a sheet of filter paper for 36 strips, litmus paper, large test tubes, small test tubes, calibrated transfer pipettes, wooden applicator sticks, and small spoons. You will also need foods such as a potato, apple, banana, cheese, or bread, 3% hydrogen peroxide, table vinegar, red cabbage, lemon juice, shampoo, and milk, gloves and goggles, scissors, and baking soda. Activity 1. What foods contain starch? 1. Place 1/8 teaspoonful of starch on a piece of paper. On another piece of paper, place 1/8 teaspoonful of sugar. Add 1 drop of iodine to both the starch and the sugar powders. A blue color formation indicates the presence of starch. Which sample changed color? Check for starch content by adding a drop of iodine to various vegetables and fruits, such as slices of potato, yams, apples, pears, carrots, celery, banana, oranges, bread, rice, or pasta, and any other foods that may be available to you. Which of the foods contains starch based on the appearance of a blue color? Record your results based on your observations. Activity 2. What foods contain sugar? Place 1/4 teaspoonful of sugar in a large test tube and the same amount of starch in another tube. Label each of the tubes as sugar or starch. Add about 5 milliliters of water to each test tube. Cap the tube and invert it gently to dissolve the sugar or starch. To avoid bubbles, do not shake the tube. Add 1 pinch of dry yeast to each of the 2 test tubes. Cap the tube and invert the tube to mix. Do not shake. Loosen cap and observe bubble information after 5, 10, 15, and 30 minutes. Which of the test tubes produced bubbles? Repeat the experiment by increasing or decreasing sugar and/or yeast. Is there a change in the bubble formation? Why is there a change? To test various fruits and vegetables for the presence of sucrose. A. Place small pieces, about the size of an eraser, of fruit or vegetable in a large test tube. Label the tube. B. Add about 5 milliliters of tap water. C. Use 2 applicator sticks to mash the food for about 2 minutes. D. Let the sample settle for 5 minutes. E. Use a transfer pipette to remove the supernatant or liquid part to a small test tube. Label the tube. F. Repeat steps A through E for 2 additional fruits or vegetables. G. Repeat step 3 to test for sucrose. Which of the test tubes produced bubbles? Activity 3. Will cooking a potato stop it from bubbling? Reminder: wear gloves and goggles. Fill 2 small test tubes with 1 milliliter of 3% hydrogen peroxide. Expose a small piece of potato about the size of a pencil eraser to high temperature by boiling it in water for 10 minutes, or a microwave in a small cup of water for 2 minutes. Place the boiled potato in the first test tube containing the hydrogen peroxide solution. Observe the bubble formation. Place a fresh piece of raw potato about the size of a pencil eraser in the second test tube containing hydrogen peroxide. Observe the bubble formation. In which of the 2 experiments did you observe lots of bubbles? Why? Experimentally determine what other fruits or vegetables will have catalase activity. Wash the test tubes and reuse. You can test for catalase activity by immersing small pieces of the vegetables or fruit in a solution of hydrogen peroxide. Compare the results you obtained and record them in your notebook. Activity 4. Can a cabbage tell the difference between an acid and a base? You will make strips 1/4 inch by 2 inch from the filter paper provided in the experiment kit. Rub a leaf of red cabbage on both sides of a piece of filter paper. The paper should pick up some color from the red cabbage and will form a cabbage pH paper. Prepare 5 to 10 additional paper strips in the same manner. Using one of the cabbage pH papers, test various items provided by your teacher for acid or base content. An example of basic solution is baking soda that can be prepared by adding 1/2 tablespoon of baking soda to 1/4 cup of water. For an acidic solution, add 1 tablespoon of vinegar to 1/2 cup of water. Compare the cabbage paper results with that of the commercial grade indicator paper provided. Which foods are acidic, or turn the indicator pink, and which are basic, that turn the indicator blue, as detected by the litmus paper? Repeat step 1, using blackberry, raspberry, or beet juices to make the pH paper. Which of these are effective to determine pH? In this experiment, you have discovered that pigments from red cabbage and beets can be used to determine an acidic or base substance, that potatoes have the enzyme catalase, and that sugar enhances the growth of organisms. Students can use what you learned to find out what other foods contain similar substances.